For this introduction, I will explain briefly about Pseudomonas putida and why is it important in bioremediation. Pseudomonas putida is a type of gram-negative bacteria that live in soil and water. It is very well studied in microbiology because it can break down harmful chemicals in the environment. This bacterium has a strong metabolism, meaning it can use toxic organic pollutants as its energy source. So instead of being poisoned, it actually grows by consuming the pollutant. It is also very flexible and can survive in polluted soil, which makes it useful for environmental cleanup. It has been used in many bioremediation projects, especially for soil contaminated by industrial chemical, oil spills, and pesticides. Overall, Pseudomonas, Pseudomonas putida is one of the main microorganisms used to store polluted soil in a natural and eco-friendly way.
So the characteristics of Pseudomonas putida from the morphology, which is physical form, it has shape and size, which is cells are typically straight or slightly curved rods, measuring about 0.5 to 1.0 micrometer in width and 1.5 to 3.0 micrometer in length. So cell dimensions can vary slightly depending on culture age and conditions. The arrangement, they usually occur as a single cells, so though some end to front pairing may be observed. The motility Pseudomonas putida is a multibacterium, so its movement is facilitated by the presence of one or more polar flagella located at one or both ends of the cell. So the pigmentation as part of the fluorescent Pseudomonas group, it produced the yellow-green fluorescent pigment pyroverdine, which is an iron-chelating slide rophore. So from the anatomy, which is cellular structures, external structures, flagella, okay, it has flagella. The primary structures for swimming, motility. So they are complex protein filaments driven by a motor embedded in the cell envelope, enabling chemotaxis toward nutrients or plant's root. So the pili or fimbriae, hair-like proteinaceous appendages on the cell surface that aid in initial attachment to surfaces and host cells, a crucial step in colonization and biofilm formation. And the last one, exophthalmosaccharides (EPS). The bacteria can produce various EPS such as hydrated matrix or slime layer, which is glycocalyx, around the cell. So this matrix helps in moisture retention, nutrient access, protection from environmental stress like desiccation and heavy metals, and biofilm development.
I'll be discussing the fascinating alkane degradation pathway, specifically as it occurs in the bacterium Pseudomonas putida. This pathway is critical because Pseudomonas putida has the ability to break down alkanes, the main components of oil and petroleum hydrocarbons, which are major environmental pollutants. It achieves this by using oxygen to convert these harmful hydrocarbons into much safer, usable compounds. This process is essentially a conversion. It starts with an alkane and ends with a fatty acid that can then enter the cell's normal metabolism. This makes it a highly important natural pathway, especially for the cleanup of polluted oil and soil following oil spills or fuel leaks. Let's walk through the specific steps of how this pathway works. Step 1, Alkene hydroxylase. The first step uses an enzyme called alkene hydroxylase or alk-beta. This enzyme converts the initial alkaline molecule into a primary alcohol. Step 2, alcohol dehydrogenase. Next, the enzyme alcohol dehydrogenase takes the newly formed alcohol and converts it into aldehyde. Step 3, aldehyde dehydrogenase. The third step involves aldehyde dehydrogenase. These enzymes oxidize the aldehyde to create fatty acid. And step 4, beta oxidation. Once the fatty acid is formed, it enters common metabolic sequence called beta oxidation. This further breaks down the fatty acid chain and the product ultimately enter the cell's central metabolism in the form of acetyl CoA. Moving on, the importance of this pathway, particularly in the critical field of bioremediation, this is where the pathway transitions from a metabolic curiosity into a vital environmental tool. I have summarized the significance of Pseudomonas putida alkane degradation pathway in four key points. The first one is enhanced soil recovery after petroleum pollution. The microbe actively accelerates the natural healing of contaminated soil by efficiently breaking down petroleum-derived alkanes. This action is crucial because it reduces the overall toxicity of the environment, which in turn improves soil quality and restores the condition necessary for both microbial and plant growth. The second is convert toxic hydrocarbons into harmless biodegradable products. Through the precise sequence of enzymatic reaction which is discussed, the bacterium transforms harmful long-chain hydrocarbons. The ultimate output of these metabolic chains is that these toxins are converted into fatty acids and eventually into a simple carbon dioxide and water. Crucially, these end products are safe, non-toxic, and easily absorbed back into the natural nutrient cycles. And the third is used in both natural and engineered bioremediation systems. The adaptability of Pseudomonas putida makes it suitable for diverse cleanup strategies. It functions effectively in natural soil environments for intrinsic bioremediation, but it also a powerhouse used in engineered cleanup setups, such as specialized bioreactors, biofilters, and large-scale oil spill treatment units. Its metabolic versatility is key to its utility in this varied system. And last but not least is highly adaptable and fast-growing in polluted environment. Pseudomonas putida possess an inherently robust physiology. It thrives in harsh hydrocarbon-rich soils, where many other microorganisms simply cannot survive. Its rapid growth rate and stress-tolerant nature allow it to quickly colonize polluted area and sustain continuous biodegradation activity over time, making it highly effectively and reliable agent for long-term cleanup projects.
What is pesticide biodegradation? Pesticide biodegradation is the process in which microorganisms convert pesticide molecules into simpler, less toxic or non-toxic compounds. In the same case, these substances are fully mineralized into carbon dioxide, water, and microbial biomass. So the common classes of pesticides that undergo biodegradation is the first one is organophosphate, such as inspiration and malation, can broken down by phosphatases and hydrolysis. The second one is carbamates, typically degrade by esterases. The third one is organocolorants such as DDT can require dehalogenesis. And the last one is herbicides such as atrazine and glyphosate, degraded through diminution, hydrolation, and oxidation. The key enzyme and their function is the first one is esterases, can break acid bonds to produce alcohol and organic acid. The second one is phosphates, can hydrolyze phosphates group and important for organophosphate degradation. The third one is dehalogenase, can remove halogen atoms like Cl and Br, a crucial step for breaking down chlorinated pesticides like DDT. And the third one is hydrolysis, can break various chemical bonds using water. A microorganism involved is a key microorganism include Pseudomonas, Bacillus subtilis, Astrobacter, Flavobacterium, and Streptomyces. can often complete degradation is not carried out by a single species, but by a community of microorganisms working together.
So there are some of key enzymes involved, which is microorganisms use several enzymes to degrade pesticides. So, for example, esterases, phosphatases, dehalogenases, and hydrolysis. And for the microorganisms involved, she is Flavobacterium, Pseudomonas, Bacillus subtilis, Atrobacter, and Streptomyces.
Hi, I will present a case study on using Pseudomonas putida for bioremediation of polluted soil. The study was conducted at an oil drilling site in Poza Rica, Veracruz, Mexico. The soil that had been contaminated over time with petroleum hydrocarbons due to ongoing drilling operations and waste management practice. I want to talk about the introduction. Mexico have a severe water, soil and air pollution problems due to contamination by both organic and inorganic compounds. Total petroleum hydrocarbons (TPH) are considered to be high priority compounds that should be removed from polluted areas in Mexico. TPH are a mix of pollutants and in the soil. This represents a risk to the health of people and ecosystem. Some of these pollutants have a low solubility in water and air, therefore difficult to remove from polluted environments. How the contamination can occur? The contamination occurs because oil drilling and maintenance activities lead to the hydrocarbon leakage into the soil. Over time, this age contamination made the soil toxic, reducing fertility and harming the local ecosystem. Natural microbial degradation is low in this condition, which makes human-assisted remediation necessary. The next slide is I want to talk about bioremediation method and the effectiveness. The researchers used a combined approach. First, they bioaugmented the soil by adding Pseudomonas putida CB100. Second, they performed biostimulation by adding nutrients like nitrogen and phosphorus to support microbial growth. Finally, they rely on the microbes' ability to produce biosurfactants, specifically rhamnolipids, which increase the bioavailability of hydrocarbons, making them easier for the microbes to degrade. This method was applied to both lab and soil microbiome conditions for realistic evaluation. Next is effectiveness. The combined treatment of Pseudomonas putida plus nutrient achieved about 61% reduction in total petroleum hydrocarbons (TPH). The production of biosurfactants helped make hydrocarbon more accessible, enhancing microbial degradation. This shows that the microbe can significantly clean up petroleum that contaminated soils under realistic conditions. The next slide is I want to talk about the advantages of Pseudomonas putida bioremediation. There are several advantages. Using a living microbe is eco-friendly. Biosurfactant production increases degradation efficiency. The method is suitable for real-age
polluted soil, not just lab media, it is also potentially cost-effective and sustainable for larger scale application. The next slide is I want to talk about application and community or environmental impact. This method has practical applications for natural resources management. Polluted lands, especially near oil industry zones, can be rehabilitated for agriculture, habitation, or ecological restoration by reducing soil and groundwater contamination. The technique protects ecosystem and local communities from long-term exposure to hydrocarbons. Compared to chemical or mechanical cleanup, this approach is more environmentally friendly and less disruptive.
The conclusion: Pseudomonas putida is a highly effective and eco-friendly agent for bioremediation, but it is also a biological engineer of polluted soil due to its remarkable metabolic diversity, its ability to degrade hydrocarbons and complex organic pollutants, as well as its adaptability to various environmental conditions and provision of effective and eco-friendly solutions. It offers a sustainable alternative compared to chemical or physical remediation methods, making it a cornerstone in modern soil restoration strategy. As environmental challenges continue to grow, the use of microbes such as Pseudomonas putida will be essential for sustainable soil restoration and ecosystem health. Thank you.